Thank you, Chris-san. And first of all, I'd like to thank to all the organizers, Himeoka-san and Chris-san, to give me an opportunity to give a talk in the nice symposiums. So I'm Naohiro TERASAKA from the Institute of Science Tokyo, so former Tokyo Tech, the Earth Life Science Institute. So I'm a wet lab biologist, but I'm also working on the evolution in the wet lab laboratory evolution techniques. And my target, the evolutionary target, is a virus-like protein complexes. It's not natural viruses. So I'd like to start the introduction about the natural viruses. So as you know, based on the modern life science definition, virus is not defined as a life. But a virus is the entities. It's really similar to the life-like molecular systems. So although the viruses have been found in nature, and these viruses have the really complicated structure, and then all the viruses are classified to the several groups based on the type of the genome or the type of the size or the structures. But the common structure is the viruses has a protein capsule in the encapsulating the RNA or a DNA genome. For example, these viruses have the DNA genome, and these viruses is RNA genome. And some of the viruses have the lipid membrane envelope around the protein capsid like this. So I think the most popular example is a SARS-CoV-2 is enveloped viruses. So also this one is not viruses, but it's like a virus-like RNA. It's called the viroid. Its viroid is just RNA without the protein complex and protein cages, but it can infect the cells and it can be replicated, hijacking the cellular mechanisms. So as shown here, the virus is really smart molecular systems, but there is no good hypothesis about the origin of viruses. One of the most popular virus hypothesis is the escape hypothesis. In this hypothesis, the virus-like protein complexes firstly emerged in the primitive cells, and once these protein complexes encapsulating the RNA or DNA genome and escape from the primitive cells, and then viruses have been evolved to the natural modern viruses. But to support this kind of hypothesis, so I choose a wet lab bottom-up approach. So this means that I'm now trying to construct the virus-like protein and RNA complexes from the non-viral materials. So this is an example of the modern viruses. So this is a SARS-CoV-2, and this is one of the smallest viruses like a satellite tobacco mosaic viruses. And also, this is a simulation movie. So one of the most simplest viruses is RNA viruses. So in this simulation movies, red colored ribbon is RNA and blue colored pentameric structure is a protein. So once protein and RNA were mixed in the solution, so these protein and RNA can be automatically assembled to form this kind of virus-like particles. So as I said, my purpose is create the virus-like structure from non-viral materials. So I firstly choose as a starting non-viral material as a rhomazine synthesis is a bacterial self-assembling protein cages as shown here. So this is the structure of the rhomazine synthesis called the AARS. So natural AARS is a 16 protein cages forming about a 70 nanometer diameter. So this is a pentameric unit of the AARS, and also this is a negatively stained the TEM images shown here. So to append the virus-like ability to this non-viral protein cages, firstly, I started to mimic the virus protein structure. So this is a virus structures examples. The one of the common feature of the RNA viruses is a positively charged N-terminal sequences on the proteins. So as you know, the DNA or RNA genome has a super negatively charged. So this means the negatively charged RNA genome can bind to this positively charged region of the virus proteins. So to mimic these structures, firstly, I append the positively charged RNA binding peptides, lambda N peptide, to the aromatase synthesis, and I also append the partner RNA sequences to the messenger RNA of this engineered aromatase synthesis. When we express this engineered lambda N peptide fused aromatase synthesis in E. coli, the structure of the protein cages were dramatically changed. So as you can see, the Aries wild type shows a 70 nanometer super homogeneous structure. With this, after the appending of the RNA binding peptides, the structure was expanded. And I extracted the RNA from this expanded structure, and I found a certain level of the small size of the RNA was encapsulated in this nucleocapsid. But unfortunately, this nucleocapsid cannot package their own genome. So because the messenger RNA encoding this protein sequences around 1,000 nucleotides, so it's too large to be encapsulated in this first, the prototype of the nucleocapsid. So I decided to move on to the directed evolution to improve this RNA packaging efficiency to mimic the natural viruses. So this is truly a wet lab approach. So this is a cartoon structure to conduct the directed evolution in wet lab. So I use the E. coli cells to express the nucleocapsid. Firstly, we transform the E. coli cells with a plasmid encoding the nucleocapsid sequences. And as I said, so in the nucleocapsid sequence, messenger RNA sequences, I appended the box B. It's kind of the binding motif to the nucleocapsid protein. And then nucleocapsid protein firstly translated from this messenger RNA, and this nucleocapsid protein can recognize this box B sequences and package the messenger RNA encoding the full sequence of the nucleocapsid. And after the nucleocapsid expression, we extracted this nucleocapsid and purify and treat this nucleocapsid with nucleus to decompose the unpackaged or the partially packaged messenger RNA. So by using this strategy, so we can selectively recover the messenger RNA encapsulated in the nucleocapsid. And this packaged messenger RNA encodes the protein sequences which can package their own genome. So after the extraction of the messenger RNA, so we converted the messenger RNA to the DNA by RT-PCR, and we repeated this cycle to do the directed evolution of the nucleocapsid. So to make the long story short, so I just showed the cryo-EM, single particle reconstituted structure of the evolved nucleocapsid. So this is the first parent aromatase synthesis. It's a 60-mer capsid. And then after the appending the RNA binding peptide, so this structure was expanded to form the 120-mer size of the nucleocapsid. And by introducing the random mutation and after the directed evolution, this capsid was evolved to form the 180-mer capsid. And then after additional directed evolution produced this NC3 or NC4, the third or fourth generation of the nucleocapsid, forming the 240-mer nucleocapsid. The one of the interesting point of its structures. So based on the early generation of the nucleocapsid, consists of the three the pentameric unit, as shown here. Firstly, monomer forms a pentamer, and the 36 or 24 or 12 pentamer forms this nucleocapsid. But the last generation, NC3 or NC4, the super dramatic structure change occurred due to the mutations, and to form the domain-swapped trimer, and then forms a 50-mer, and then forms this like a sucker ball-like structure. And finally, this NC3 and NC4 can package about the 2 copies of the messenger RNA into the 1 capsid. So this means that RNA packaging efficiency is really comparable to the natural viruses. And recent years, we are conducting the further engineering the evolution of this type of the nucleocapsid. So by using the protein ligation and split capsomer and circular permutation. So this is the purpose of this engineering is to append the more complicated functions to this artificially evolved nucleocapsid to mimic the natural viruses. And one of the ability of the natural viruses is infecting to the cells. So to append the infection ability to this artificially evolved nucleocapsid, we developed the protein surface modification systems by using the protein ligation tag. It's called the dog tag and the dog catcher. So we co-express the protein capsid modified with a dog tag, and we also co-express the functional motif fused with a dog catcher. So by using this bicistronic messenger RNA system, we can easily express this surface-modified nucleocapsid. And after the several rounds of the directed evolution, finally, we developed this surface-modified nucleocapsid. And by using the surface-modified nucleocapsid, we additionally further appended the receptor binding motif by using this lasso grafting technology. It's based on the cyclic peptide. So anyway, so after appending the receptor binding ability to this surface-modified nucleocapsid, we further conducted the directed evolution using cell lines. So in this system, so we firstly express the library of the surface-modified nucleocapsid, and this nucleocapsid library were added to the HeLa cells. And after treatment of the nucleocapsid, we can recover the cell lines bearing the delivered RNA from the nucleocapsid, and then perform the RT-PCR and ligate the DNA to make a next generation of library. So by repeating this process, the RNA delivery or the cellular infection ability was improved. So engineering, the ability was improved about 2.6 times, and further additional directed evolution and ability was improved about more than 5 times. And then finally, so this is a construct of the finally evolved nucleocapsid can infect to the HeLa cells. And this is a fluorescent microscopy data. So red color means the delivered RNA. So this is this image shows the RNA was delivered, and then nucleocapsid can infect to the HeLa cells. And when in the presence of the MET inhibitor is a target receptor inhibitor, the delivery efficiency was dramatically decreased. So this indicates that this surface-modified nucleocapsid can infect the cell via the receptor-mediated endocytosis. And another strategy to modify this nucleocapsid is protein splitting. So this is a protein, the NC4 protein illustration, and we split this protein monomer to the N-terminal fragment and the C-terminal fragment, and we express these 2 fragments in the E. coli. And surprisingly, even after the splitting of the protein unit, this nucleocapsid forms almost the same structure to the non-split variant, like shown here. This is a non-split variant, and the bottom one is a splitted variant. And only the difference is RNA packaging efficiency. As shown here, this blue bar indicates the copy number of the messenger RNA packaged in the nucleocapsid. In the case of the NC4, more than 2 copies of the messenger RNA was packaged, but the split NC4 there is almost just one copy of messenger RNA were encapsulated, but it's almost the same as the natural viruses. And by using this split nucleocapsid, we are trying to mimic the virus-like surface chemistries. So the first example is a sugar modification on the surface of the nucleocapsid. So in the case of the natural viruses, have the sugar modification on the surface of the particle to escape from the immune systems. So in the case of our project, so we co-express the sugar modification enzyme, and we append the sugar modification tag to the split nucleocapsid, and we confirm the almost 100% nucleocapsid was modified with the sugar. And also the second example is antibody display to append the cellular targeting ability. In this case, antibody binding domain was displayed in the split nucleocapsid, and we can freely modify this nucleocapsid with any antibody. So due to time limitation. So also, so we are trying to control the size or the structure of the artificial nucleocapsid. So in this case, we developed the in vitro reconstitution system of the nucleocapsid. And by changing the cargo molecules, this protein structure was dramatically changed. So for example, in the case of the RNA packaging, the nucleocapsid structure is almost similar to the E. coli expressed nucleocapsid, but when using the structured DNA origami, like a sphere-like structure and rod-like structure, we can control the protein assembly structure as shown here from like a big sphere or the rod-like structure. And finally, so I'd like to show the really preliminary data to you. So as I explained, so we are working on this type of the nucleocapsid, but we introduce another type of the circular permutation and the directed evolution. Now we are trying to make a different type of the structure from the sphere-like structures. So we conducted another directed evolution to the novel circular permutation variant. Finally, we obtained this construct showing the super heterogeneous structure. Also, now we are working on the cryo-EM analysis, but recently we found a super unique structure in this particle. So it's like a 42 pentamer. It's not sphere-like structure. It's how to say it's like a rugby ball-like structure. So share here. So anyway, so finally, so I'd like to thank all my lab members and collaborators. So because I started my laboratory about 4 years ago, so now I'm recruiting the graduate student and the postdoc. So if you're interested in this kind of the wet lab directed evolution technology, so please let me know. So thank you for listening.
Any questions or comments? We have 2 minutes and a half. Yes, please.
Yeah, thank you for this display of engineering. I was also curious. So, do you also test if they're still infectious? Do they still infect the HeLa cells or the HEX cells? And does the efficiency change a lot with, for example, taking a split version versus a circular permuted version versus for all these different structures?
Okay, thank you so much. So far, the answer is I'm not sure, honestly, because the infection ability is totally depends on the type of the receptor and the affinity of the receptor of the targeting moiety. So I'm not sure. But the assembly efficiency, this means the nucleocapsid forming efficiency is totally depends on the circular permutation or the split variant. But to our experiences, the split version is really good. To form the nucleocapsid particle, it's really comparable to the non-split variant. So it's really surprising me. So, but after appending or after the modification of the surface, so the assembly efficiency was dramatically decreased. But in our cases, another additional directed evolution can improve the assembly efficiency by introducing the several mutations. So.
But could it also be that there's a limit to, because if the efficiency of assembly is too high, I mean, in the cell, it also needs to open up again. So is that something that is also an issue or not?
Yeah, it's kind of the balance between the assembly efficiency and the disassembly efficiency. Yeah, now so yeah, we are working on this kind of topics. Yeah.
Interesting. Thank you.
So any short comment or short questions? Okay, so, oh, yes, please.
Thank you very much for a nice talk. Maybe I'm missing some part, but do you have any idea? So what is the origin of this virus protein? This virus protein, maybe the originally came from the host cell DNA or RNA. So
Yeah.
why the host cell have this structure?
Yeah, good question. So based on this escape hypothesis, so the first is cellular protein have to assemble to make this kind of particle structure. But in the case of the modern cells, even in the modern cells, this kind of protein cage-like structure was found. For example, in the case of the enzyme cascade, this kind of the protein assembly is really beneficial. So I think so to this hypothesis, firstly the cells form the protein complexes to for another purpose, like to improve the enzymatic activity. And then sometimes such kind of the protein assembly gets the ability to package the genome information. So that is this protein virus escape hypothesis. Yeah.
Okay, so let's to speak again. Of life will be start at 1 o'clock. So enjoy your lunch and please come back